The act of fertilization plays an important role in the maintenance of the life cycle. It is an extremely complex cellular process that is responsible for the creation of social entities and the initiation of an individual's life. In mammals, the main characters for this act are the female's egg cell and the male's sperm cells, which will fuse to form a zygote. This zygote will develop into an embryo and finally into a newborn child after 9 months of pregnancy. In this video, we will elaborate the process of mammalian fertilization at cellular level. And trust me, it is a lot more phenomenal than we thought. The process of fertilization begins when the male ejaculates his sperm into the female's vagina. These freshly ejaculated sperm are still considered as imperfect or immature and cannot or will only partially fertilize the egg cell. Thus, they must first undergo a series of processes known as translocation and capacitation.
The female reproductive tract is highly specialized to actively regulate the transport and maturity of both the egg cell and sperm cells. The translocation of sperm from the vagina to the oviduct involves several processes. In translocation, the muscular activity of the uterine muscle contraction will help the sperm to aggregate and to achieve motility that will allow them to reach the uterus faster. The sperm will then swim against the flow coming from the oviduct to the uterus, which is known as sperm rheotaxis.
After spending some time in the female reproductive tract, sperm will undergo maturation and acquire competence as they reach the ampulla. However, these properties may be lost if they spend too much time there. This is where capacitation takes place. Capacitation allows the sperm to acquire the ability to penetrate and fertilize an egg through a series of biochemical events that involves a number of enzymes and signaling molecules to remove its adherent seminal plasma proteins. Capacitated sperms become hyperactivated and destabilize the sperm's membrane to prepare it for the acrosome reaction. The final stage of sperm migration is chemotaxis. Oocytes and its cumulus cells secrete hormones such as progesterone that attract capacitated sperm towards the egg cell.
In order for fertilization to occur, the sperm must first penetrate the layer of corona radiata that surrounds the egg cell to reach the outermost layer of the egg, the zona pellucida, where they will attach to a species-specific sperm receptor that trigger the acrosome to release its digestive enzyme, acrosin, that allow the sperm to penetrate the zona pellucida, but only the fastest sperm can fuse with the egg cell. This event triggers the release of chemicals from the cortical granules that destroy the sperm receptor, which is the binding site, and harden the glycoprotein matrix of the zona pellucida in a process known as the cortical reaction, preventing further sperm entry that may result in polyspermy, which ensures that the zygote form is diploid. Now, the cilia and the fallopian tube will sweep the zygote towards the uterus, where it will be implanted in the uterus lining. This newly formed zygote will develop and begin the life of a new human being.